Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the isotypes or the classes of the antibodies. In the last video, we were talking about the important properties of a subclass of immunoglobulin G, which was known as the immunoglobulin G1. In this particular video, I want to focus on the important immunological functions that the immunoglobulin G1 can play. So the first immunological function of the immunoglobulin G1 is that it is an activator of the complement system, and it is a very strong activator of the complement system. Now, if you talk about the immunoglobulin G1, what they do is that in activating the complement system, they are going to bind to the C1q component of the complement system. And the C1q is the first component of the classical complement pathway. We have already discussed in detail about the classical complement pathway, and there I told you that the C1q, that is the first component that the classical complement pathway use for the activation. Now, if you talk about the C1q, where the C1q it binds to the immunoglobulin G1. Now, the heavy chain that contain a different constant region, and we call it as the constant region 1, constant region 2, or the constant region 3 of the heavy chain. So the C1q binding site that is available in the second constant region of the heavy chain of the immunoglobulin G1. Now, why the C1q? It binds at this particular position. And the reason is that there are some key amino acids like the tyrosine 296, the glutamic acid 318, the glutamine 322, the protein 331, the aspartic acid 337. Now these key amino acids, they are actually providing a strong binding site for the C1q and hence the activation of the classical complement pathway. And these specialized amino acid is one of the key reasons that the immunoglobulin G1 is a strong activator of the complement system in comparison to the other classes, other subclasses of the immunoglobulin G, like the immunoglobulin G2, 3, and 4. So this is one of the reasons that the immunoglobulin G1 is a strong activator of the complement system. Secondly, if you look at the glycosylation level of the immunoglobulin G1, so there is a lower glycosylation level in the FC region in comparison to the other subclasses of the immunoglobulin G, like the immunoglobulin G2, 3, and 4, thereby making it a strong activator of the complement system. So this is the first immunological function that the immunoglobulin G1 it can perform. And we have already discussed in the classical complement pathway that when that activates, how it is going to neutralize the foreign pathogens. The second important immunological function that the immunoglobulin G can perform is that it is a very strong activator of the antibody-dependent cell-mediated cytotoxicity. We will have a detailed video on the antibody-dependent cell-mediated cytotoxicity, but at this particular time, to understand the ADCC function of the immunoglobulin G1, I'm just going to define what is ADCC. So ADCC is actually a process by which the antibodies they mark the harmful cells, and then the immune cells they are directed to kill those particular cells. So you can simply say that the immunoglobulin G1 is actually tagging the foreign pathogens to be killed by the other immune cells. Let me give you some examples of this. When we talk about the cytotoxic cells, the cytotoxic immune cells, so the natural killer cells and the macrophages, they are the main player. And on the surface of the natural killer cell, this is an electron emit of the natural killer cells. So on the surface of the natural killer cells and the macrophages, you are going to see specialized receptors, which are known as the FC receptors, specifically the FC gamma R3, which is also known as the CD16 receptor. So you are going to see this particular receptor on the surface of the natural killer cells and the macrophages. Now, what happens is that if you look at the structure of the antibody, here we have got the antigen binding site. So, these antigen binding site, they are responsible for binding to the antigen. And of course, in this particular case, we are talking about the immunoglobulin G1. Now, if you talk about the Fc receptor binding site, so that is available in the lower hinge region, and the second constant domain of the heavy chain, that is actually providing a binding site to these Fc receptors which are present on the natural killer cells. So the natural killer cells or the macrophages, they are going to bind to the immunoglobulin 1 utilizing its Fc receptors, and the Fc receptors, they are actually having a binding site for them in this particular area, the constant region 2 and the lower hinge region. Now, let me give you an idea how this particular mechanism of the ADCC works. So, the antibody, they are going to bind to the antigen on the surface of the target cell. For example, if this is a pathogen, the immunoglobulin G1, they recognize specific receptors on the surface of these pathogens, so they bind over there. Now, once the immunoglobulin G1 excuse me, once the immunoglobulin G1 that has bind on the receptors on the surface of the pathogen, they are then providing the binding site to these natural killer cell or the macrophages. So the macrophages or the natural killer cell, they are going to use their CD16 receptor to bind to the lower hinge region and the CH2, the second heavy chain of the gamma chain. Once they bind over here, what they actually do is they are going to go for the degranulation. And when the natural killer cell or the macrophages, when they go for the degranulation, they are going to release important components. The first component they are going to release, that is known as the perforin, and these perforin are responsible for making pores in the target cell membrane. And in this particular case, of course, our target cell, that is a pathogen or that can be a tumor cell. Secondly, once these perforin will make pores in the target cell membrane, the second component that the natural killer cell are going to release, the granzymes. These granzymes are then responsible for the activation of signaling pathways that leads to apoptosis. And once the apoptosis that has been performed, you have removed the damaged cells like the cancer cells from the system. So the tumor cell, they are going to die by the process of the apoptosis. When we talk about the pathogens, and say for example, if we consider this as a pathogen, then the perforin, after making the pore in the target cell membrane, the pathogen, they are going to lose important components from their cell, which can actually lead to their death. When you talk about the human cells, for example, you are going to kill them by the granzyme, which activates the apoptosis, thereby leading to the removal of the cancer cells from the body. We will continue our discussion in the next video.